Annie Liu and Sunanda Sharma. Could you two join me on stage? Awesome. And I'm going to do a quick just reading of your bios, if that sounds great. So Annie Liu is a research-based artist whose work examines the reciprocal relationships between science and technology and their influence on human subjectivity, culture, and identity. She is passionate about integrating multidisciplinary approaches to art making and is currently an associate professor of practice at the University of Pennsylvania. Sunanda Sharma is a creative biologist and interdisciplinary scientist interested in questions of astrobiology, evolution, ecology, biodiversity, origin of life, and artificial life. She completed her PhD and master's at the MIT Media Lab as part of the Mediated Matter Group and is now a research affiliate with the MIT Space Exploration Initiative. Thank you both for joining us. Take it away. Make sure you unmute yourselves.
Thanks, Veena. Hi, Sunanda. It's such a pleasure to be speaking with you on the stage today. Like Dan, I just wanted to take a moment to acknowledge the really awful news in the US today with abortion rights being overturned. But I also wanted to take a minute to congratulate all of the participants in the Biodesign Challenge because I had the pleasure of being one of the judges, and the range and the depth of research, the type of projects that you are doing is so incredibly inspiring. So I have hope for change to come and lots of futures that are different than the ones that we're living in today. I wanted to introduce our panel by saying that we are both designers, artists, researchers that work across a lot of disciplines. I met Sunanda first at MIT Media Lab, where in the very first impression, I was so intimidated because Sunanda is so smart and so has amazing educational pedigree and and then you meet Sunanda, she's just the kindest, most wonderful, best hope for a collaborator you can ever have. So I just wanted to open with that. I thought maybe we could start by talking a little bit about our backgrounds for some of the audience members who might not know anything about our practice. And then I've prepared a list of questions that I tried to grab on Instagram, asking people what they want to know about our experiences with collaborating across disciplines. Would you like to start, or would you like me to start, Sunanda?
You go ahead. I like listening to you.
Okay. So hi again, my name is Annie, and I am an artist, researcher, and designer. And to give you a little bit of background, my undergraduate degree is actually in art. I studied art at Dartmouth College. It was a liberal arts education. And I love that because it forced me to take classes in spaces that I was very uncomfortable with. I have to say that I grew up at a time where people, even teachers, would still say things like girls are not good at math or girls are not good at science. And somehow I really internalized that and became really intimidated by certain subjects. But I think in a way, by being forced by my liberal arts education to take a really wide variety of disciplines, I learned that, actually, I can learn these things, and especially through the lens of art, by having a certain kind of desire for a project, I would then almost like naturally learn certain things. And then I went on to architecture school, actually, because I came from a relatively low income family, and I couldn't imagine what it could be as a career path. To be an artist, it seems so impossible and mysterious. So I studied architecture, which I really love. It introduced me to digital fabrication. And also it caused me to think about the context of making outside of the thing itself. So for instance, when you're an architect and you design a hospital, for instance, you learn a lot about the physical building, the HVAC systems, the electric systems in the building, the user interface in terms of like the materiality that might help someone heal or feel comfortable. But you also have to learn about the governmental context in which this hospital is situated, in the historical context as well. So it caused me to situate all of my projects in a larger scheme, really taught me to think critically. And then I had the pleasure of also having a Master's of Science from the Media Lab at MIT, where I met Sunanda, and I studied with Sputniko there in a lab called Design Fiction. And I think that's where I really doubled down on learning more about how to think critically through design. So all of our projects there are through, actually, it happened so that while I was there, all of our projects were within the domain of biodesign. And I would say a main focus of my group was in feminist technoscience. So thinking a lot about biopolitics. Actually, my thesis project was about bodily control. My very last year at the Media Lab was when there was an election where we elected Donald Trump in the US. And he infamously said this phrase, grab them by the pussy. And there were a lot of protests in which protesters were wearing or holding signs saying the pussy grabs back. And I happened to be in a class with David Kong called Grow Almost Anything at the time. And I was learning about something called galvanotaxis, where the researchers at Stanford were engineering paramecium to swim directionally with electric fields. And I was wondering, does this work with sperm, and what does it mean for a woman to control sperm, given the entire history of men controlling the reproductive systems of women in biopolitical regimes? And so the piece is called Mind Controlled Sperm. You're welcome to look it up online. And yes, and I'm sorry if my brain is really scattered today. There's just a lot of emotional processing to do in this kind of political context. And so, yeah, that's a little bit about me and the journey that I took to get here. Sunanda, do you want to talk a little bit about your background?
Sure. Yeah. So thanks for having me. First of all, this is so exciting because I think I've thought a lot about biodesign, and I always feel like I'm learning as I listen to new projects. And when I was looking at the projects that people are presenting, and when I was helping out at Mass Art this year as sort of an expert scientist and talking to different students, I was just struck by how much hope there is there. And I think it's really profound, and how much awareness there is to think outside a particular prescribed way. And I find that really promising. So it's a pleasure to be here. And it's a pleasure to be here with Annie, because we are actually friends that we met years ago, and being together is wonderful, even today. So yeah, I took a similarly winding path through a lot of different fields. And someone told me something very similar to Annie, that I was failing out of all my classes at MIT for like the first 3 years I was there. And they were like, oh, well, you should, talk to different people about this, talk to deans, talk to mental health, things like that. And someone told me, well, some people's brain just doesn't work that way, that you can do coding and math. And I was like, well, that's unfortunate. Sounds like I'm one of those people. And then another dean, just one person out of so many, actually had something useful to say, which she said, hey, maybe you're not good at tests, but you're great at projects. Sounds like you really like research. Just do that more. So as soon as I switched to that, my grades went from like alphabet soup of C's, D's, and F's going all the way up to A's and B's, because it turns out just doing research in a really unstructured way that's where my passion is. As a kid, I wanted to be a poet and a naturalist, and I liked finding organisms, and my mom was really upset at me for bringing them home, but I'd always bring them back out, but still she was upset at me. And I ended up getting an undergraduate degree in biology at MIT, and I worked for almost all the years that I was there in a neuroscience and neurotechnology lab looking at optogenetics, so turning on and off brain circuits and understanding fear and memory circuitry, like spatial memory formation. So that was my first great experience of what it's like to be in a lab. And then I wasn't really sure what I wanted to do. And through a random email chain and some chance meetings, I ended up meeting Neri Oxman at the Media Lab and starting at the master's program there. And I went from being surrounded by biologists, physicists, and neuroengineers to being the only biologist in a group that had all architects and designers and a couple mechanical engineers. And this was a huge shift in community, but also in perspective for me. And to use Jennifer's word, this is when I became a hybrid person, officially, at least. So that was a huge change in terms of language, in terms of currency in a way, and in terms of just what outputs or goals are desired for the projects that we're doing. And communication was really hard for me to get people to understand sort of my side of what was happening, because I felt very intimidated the other way by all the designers, that everybody just knows all these references and they can talk about this one exhibition that happened this one time in the Serpentine Pavilion this one time and just all know about this. I'm so behind. And someone gave me the advice of just look at stuff that you like and try to think of why you like it, what it makes you think of, what the context of when it was built, and just start there from that very genuine place. And that was really good advice that I come back to all the time. And I continued with my PhD in the same department, and I tried to continue weaving this sort of path. And I'll just note this, and I'm sure we'll talk about this in the questions, but this question of what's rigorous and what is accepted as a hard science or technical work, I think is something that I've always tried to think about and challenge, because the number of people who told me that I was throwing away all my potential as a scientist by moving more into design was very high. That I was going into fluffy work and I wouldn't be able to do anything. And so being in biodesign it feels to me like defiance, and I use that anger a lot, I think, in my work, because I think science is very rigid in a lot of ways and it doesn't need to be. And being in this group of all these other people who are thinking different ways allowed me to question, why do we culture E. coli at 37 degrees? Yeah, it's optimal for the rate of growth, but like, who said that? And why are we all passing that down? And if they are actually changing, like this lab strain is still changing over many, many, many years, should we still be doing that? And also, what are we optimizing for? If it's for us to have ease of culturing, why? Should it be something else? Should we optimize for diversity? Should we optimize for organism comfort? Can you do that with microbiology? What are the bounds of empathy between us and microorganisms, which also make up us? So there's a lot of different things in there that I tried to engage with. And a space that I like to do that was this biodesign lab that we built. So I co-designed and built this during my master's back in 2015, 2016. So it's a BSL2 lab that's in the Media Lab now. And it's a place where we did pretty hardcore microbiology, but we also had a lot of art and design that was happening there. Like we had an artist do a performance in there one time, and we also created a lot of biohybrid objects that eventually got exhibited at different museums. So some of my work or most of my master's and PhD work was at MoMA in 2020 as part of Mary's exhibition, and more recently just wrapped up at the Nature X Humanity show that was at SF MoMA earlier this year. And so I had the exhibition thing, and then also a first author paper in a scientific journal presented at academic conferences. And my committee was Neri, who's an architect, designer, and many other things, inventor as well. Another inventor and computer scientist and electrical engineer. And then my undergrad advisor, who's a neurotechnologist and neuroengineer. So that was a really big mix for me. And I ended up also getting excited about extreme environments. So I got to lead a project that was aboard the ISS, the International Space Station, for 30 days. And now I'm an astrobiologist officially, and I'm still in love with design. So a lot of other stuff happened on the way, but that's the brief overview.
Amazing. I hope everyone out there is swooning like I am. I actually had a whole bunch of other questions, but I wanted to start by maybe expanding on something that you said. How wonderful that you met a dean that was like, maybe you're more of a project-based person than an exam-based person. And I wanted to bring that out because sometimes when we do our projects, there are tricky technical things to learn and to, I don't want to say overcome, but sometimes there are hurdles in creating a project. And when I talk to my own students, sometimes they'll say like, I'm not a coding person, or I'm not a very math-based thinker. And I always like to give the analogy of like, maybe it's just that in traditional math curriculums, they try to teach everything as if you are a square peg, but really you're a triangle. And like, you're just trying to find the shape that fits in your knowledge puzzle. And so I always try to present information in a variety of formats. So maybe you're a visual thinker, or maybe you're a verbal thinker, or maybe you're a symbolic thinker. And let's try to communicate the same thing in many different ways. And I feel like the work of biodesign and bioart inherently does that, because you take, you have to be kind of like a multi-language speaker in a way. And I was wondering if you might expand on some of your experiences with that, especially if you are, I don't know if I'm overusing this analogy, if you say work and think in triangles, but you're collaborating with someone who works and thinks in circles, for instance. Like, what are some, do you have an example or memories of that kind of translation process?
Yeah. Oh, that's a good question. I think this is something I struggled with so much when I started at the Media Lab, because I would say stuff like, we would have this conversation, which was just like, the designers would say, what can the biology do on this project? Like, it's going to be a thing that you put on top of the design, and then it just comes on top. And I'd be like, well, what can design do? And our answer for both was just anything, right? And that wasn't really productive. So the better thing to do ended up being, as you're saying, diversifying in terms of how we were presenting our work. So I didn't appreciate at all the power of visually explaining work, not with graphs and plots necessarily, but also just showing what it is, almost doing like portraits of organisms I work with. So I really love microscopy. Like, that was my first love in lab. And that was the way that I started being like, all right, this is E. coli. This is what it looks like when it's has all the nutrients it needs. This is what it looks like when it's stressed. This is B. subtilis, same sort of thing. This is human cells. And speaking that way, and now I like put it everywhere. You can see my background, some radial area. And I think that everybody could interpret images in the way that they wanted to. And so it came from this place of like excitement rather than I'm trying to get to a productive outcome immediately. So I think making some space to then start to throw around language that isn't even precise, but that explains how you're feeling and what you're seeing was way more productive. And I know, like, when you're talking about your projects, you talk a lot about like the art and the motivation, but then you also throw in words like galvanotaxis. And, like, you have that same sort of polyglot nature, I think, and you get it over time. But I think it's really hard to do without, I don't think you can do it without feeling stupid. And I think I felt stupid so often, and that's been great.
Yeah, actually, maybe to go on that, it helped a lot for me to always have the beginner's mindset. And I know it's very cheesy, but in spaces like Harvard and MIT, there is such pressure, I think, to live up to a certain myth of what other people in those universities have achieved, or like the word genius gets tossed around a lot, and imposter syndrome. But it helped me to just shed all of those pressures and just pretend like I was in a kindergarten and just be like, hey, I'm playing with blocks and that person is playing with clay, and I want to go over there and learn a little bit about the clay. So what are some like really basic entry level ways that we might play together? It's funny, maybe I'm thinking about this a lot because I have young children, and it's funny because I'm often, there's a Reddit subreddit called Explain It to Me Like I'm 5, and I'm literally always actually explain it to you. Like, actually my children are even younger than that, so I'm always explaining it to her like she's 2, which she is. And I find that it helps so much because, especially like, my background is in design, and there's a lot of design theory and lots of philosophy that uses really long, fancy words. And you'll reference people like Heidegger or Foucault, and a lot of times, a lot of the other people in the room might not know what you're talking about. And why not just say it out in plain language instead of all of these big words? So I think there's that too. I think it's tough because in both academia and fine arts, there is a lot of posturing, and I think it probably comes from a place of insecurity. And so I try to live out what I preach, which is like to stay really humble, to explain it like you're 5, and to like go back to these really basic tenets of being a good human in a way, like being someone who's just trying to, yeah.
I always felt like the anger for me was always with this gatekeeping.
Yeah.
Enough to like go into this lab and go do the project that you want to do. And what is enough? Do we want to continue the legacy of gatekeeping for science? Because it sounds to me like for generations, we've missed incredible ideas and incredible chances for exploration because we've been gatekeeping people who don't look like they should fit in or who don't have the right words to express what they're thinking. And of course, like, I think people try to use things like safety in some ways, or like almost tradition, which we say as protocol or citable protocol or things like that in science, without actually thinking why that's the case. And I think pushing that has always been really important. And that's why I appreciate, I think, as you're saying, like being really grateful and humble with people who are able to push that. And that's what I felt that I had in a lot of the interactions at the Media Lab, especially with designers and artists, like push that thing and figure out why we even think that way.
Mm-hmm. I'm looking at our list to try to make sure we hit a lot of these questions. The first two questions were actually, how do you meet collaborators? And how do you define a successful collaboration? Maybe those are hand in hand. In terms of where to meet collaborators, I have stayed for a large part of my life in academia, and that has been a really rich playground. I mean, academia has its problems for sure, but it's been a really wonderful space for me to try to meet people across really wide ranges of disciplines that I might not have normally encountered on the street. Yeah. And then the other way that I meet collaborators is by writing grants. There's, like, for instance, a really great, I don't want to call it a competition, but it's called the BAD Awards, Biological Art and Design Awards, and it's based in the Netherlands. And in that scenario, artists, designers, and scientists apply to get paired together. So you meet people who are inherently motivated to work with you.
Yeah, I think maybe I haven't been quite as formal, maybe, or no, parts of that with academia, because I've been in academia for a while. And when I graduated with my PhD in March 2020, and nobody.
Congrats! Yay! Amazing.
Sad. And it really made me start thinking, like, oh, how do I have collaborations if I'm not in academia anymore? How do I do that? And so I just basically spent a bunch of time, like, I eventually started my own little, like, one-person consulting thing and asked everybody, like, hey, do you want to talk about 3D printing and cells? I got you. Let's do it. Or like, do you want me to build a lab in my basement so I can test out your microscopes? I'm happy to do that. And so that was really exciting. And I met a lot of people in that way just by, like, reaching out by email, kind of, or having people connect me to other people. So every person in the network from academia, asking them to tap shoulders and see where to go. But I also just spent a lot of time cold emailing. Like, I read people's papers and I said, hey, you're a professor here. I read your paper. I thought it was cool. Can I talk to you about this? This is where I think our work could overlap. And I thought that would lead me to a postdoc doing science, doing astrobiology. What it actually led me to that year was doing an artist in residence. So this was a really weird changing of hats for me, because I was so used to being the scientist with a bunch of artists and designers around, and us working together in that way, but having to be, like, sort of the science expert. In this case, I was the artist in a lab of everybody who focuses on fungal biotechnology. And I did a project on can we have empathy for microorganisms, and focused on one particular type of black fungus in the lab of Vera Meyer, who's a professor at TU Berlin. And that was just incredibly fruitful, like, just joyous to work together. There was so much joy and happiness I always felt, and I was so inspired working with her and the people in her group, and also being able to give myself room to think in a different way and to push myself, like, okay, I went to technically the School of Architecture. Let me think about this. Let me try to hold myself to this standard. And so, like, I think being really bold about just reaching out to a bunch of people was the way to go, and then being open to the potential ways you could work together. And I've even met people randomly who, like, were bioprospecting for an organism, and we met on LinkedIn or something like that. So a lot of different ways.
That's awesome. Yeah, I totally forgot about that. I also totally cold email sometimes after reading. Actually, sometimes if I read a paper I really like, I just email the person as a fan. I feel like pop stars might get lots of fan mail, but I feel like scientists don't get enough fan mail. So I try to acknowledge when I really love something.
Yeah. And then in terms of like a successful collaboration, what do you think? What have been successful collaborations for you?
That's such an open-ended question, because sometimes I struggle to define what a successful project is for me, like just stemming from this. But I think for me, successful collaborations are ones that open more doors, where I get something in me sparks, and like one project leads to 10 more ideas in my studio, or I learn so much more, or something about the way I view the world. There's a paradigm shift. And I think that's true for the researchers too, because for instance, maybe to give a concrete example, I had the fortune of winning the Biological Art and Design Awards in the Netherlands in 2017, I think, or 18. And I worked with researchers from I worked with radiologists, and I asked really strange range of questions. And I remember over and over again, they would say, I'd never thought of that. That's like 10 more research projects in our lab that could be launched. Or we, being mindful about respecting the equipment and not breaking it, of course, but there were a lot of like, we never thought to use the MRI in this way. And what are some of the new potential applications that could come out of it? So I think that there are moments of great synergy. Also, I think just making a really good friend is huge success for me, because I don't know, like the world can be really dark and lonely, and finding wonderful people who bring light into your life is a huge delight and joy. So yeah, what about for you?
I think, success is a hard thing to quantify. Like there's the things that I could initially think about of like, we published it, woo, whatever.
Mm-hmm.
But I think the things that felt most exciting in terms of collaboration was when we go past the superficial level and we get to this like deeper one. Like we're speaking the same language, maybe not with the same words, but we're definitely on the same page. And I think often there's a moment of surprise that comes with it. So a project that I'm thinking of is I worked with a bunch of other folks in the mediated matter group when I was doing my PhD, and we were making these new types of death masks, essentially. And so we broke a lot of 3D printers doing it, but we were engineering organisms, bacteria, basically, to interact with chemicals that could be released from a 3D printed surface, and looking at what that looks like on the human face. What does that look like in the scale of the human body? And what does it mean in the context of the traditional death mask, which used to preserve people's likenesses, but now we have many more biotechnologies across different scales. What does this mean in terms of how we can represent humanity, humans after life, and maybe beyond death? What does that mean? And what was interesting is we used a really, really simple assay, so a type of experiment, basically. And we just used bacteria that are engineered in a way that you literally learn in like a lab class during undergrad. And we did essentially a type of blue-white screen. And it was very basic. And so the people we were working with are synthetic biologists, and they were at the MIT Synthetic Biology Center. And they're like, oh, how'd you do this? And we were telling them, well, we just expanded the blue-white screen basically to different colors. And they're like, wow, I didn't even know that. I couldn't have recognized that. And these people, like me, had been trained to work with that. But there was this big moment of surprise of like, okay, it's in a different context. It's with this death mask thing. It's on a 3D printed surface. It's interacting with the surface and sort of building little chemical microenvironments that the bacteria are working with. This is a different thing. It's what I was familiar with, but in a different way. So it has this element of surprise. And I feel like that made it feel very fruitful, because we were able to take something so known from another person's field and twist it in a way that it could be applied somewhere else. And so that was really, really interesting.
I love that. Actually, it bridges into another question I had that I struggle with in my own practice. So maybe it's an open-ended question, but I wonder what it means for you to collaborate with another species. Because I think in a lot of bio art and biodesign traditionally, perhaps even when the collaboration appears to, or is called a collaboration, sometimes it's a one-way human exploiting the other species type of relationship. And I struggle with this because I think I'm guilty of this too. And these days, without having an answer myself, I think a lot about what does it mean to really care for the organism or to really have a symbiotic relationship. It makes me think of, and maybe I am saying this artist's name wrong. I think her name is J. Rim Lee. It's a piece that I saw a long time ago. I think she did it for her master's project at ACT at MIT, the Art, Culture, and Technology program, where she changed her own diet to make her urine more nutritious for a plant garden that she was building. And so it like really blew my mind. I was like, oh my gosh, usually we modify the plant to be more tasty for us, but she's trying to modify herself to be more beneficial to the plant. And I love this. I keep thinking about it. I don't know. Do you have any thoughts on this type of interspecies collaboration?
Yeah, the longer I'm in this field, the more I think about the terminology that we use, also because of work from my amazing collaborators and colleagues, sort of pushing what does that mean, interspecies collaboration? And it comes to, I think, the question of agency as well. Like, how can you recognize agency in another species? And I've tried to think about that going all the way down to the micro scale. Like, there's mammalian sort of stuff that you can sort of see, like this organism is, you can tell if a mouse is anxious and you can tell if it's relatively calm, for instance. But can you do that for a bacterium? Is that even the right terminology? Like, calm? Because they wouldn't have the same range of potential states, maybe. So I think, like, I don't really have an answer. It's more that now I'm questioning all the terminology. Like, I think people a lot of times in biology like be like, oh, my cells are happy because they're growing really well in a way that's useful for me in my experiments. They're growing really fast and efficiently. And just like that terminology happy never sat right with me. Or like, my cells are so sad, they're all dying or something like that. I'm like, okay, how can we like, do we need different words? Do we need to apply a whole new set of terminology for this? But I think one thing that I found that was actionable, aside from changing language or questioning language or putting in caveats as I'm presenting things, was to bring this question of discomfort for the viewer, for the human. How we were doing one project where we're working with a bunch of insects and we wanted to put them into a museum setting. And I was like, okay, well, the best thing for this insect is to be at like 30 degrees Celsius and 85% humidity. So can we do that in the museum? And I mean, you can guess the response from the museum was like, no. Like, well, then why are we considering bringing them in there if we're not going to do something that makes them the most comfortable, that's most similar to their native habitat? Because maybe we shouldn't be doing that. So if we can't make viewers and ourselves uncomfortable for the sake of collaboration with another organism, then we really need to question, is that a true collaboration? Is there equality there, if that's even possible? So I think there's so much to dig in there.
Yeah, no, those are such great insights. I like to think about it too in this way, like, maybe it's not about me. Maybe we call it disobedient, but actually it's just like, different ways of being in the world. And so going back to maybe the topic of biocontrol, I think that one of the things that, and sorry to make this sound like it's closing remarks, but I think we only have one more minute.
Yeah, it's okay.
I think that one of the things that makes it so uplifting to me, the biodesign challenge, is by its nature, it is very collaborative. I see lots of students working across lots of disciplines in large groups. And I think about how important it is to be able to work together, especially in intense times like this, to think about what it means to, I don't know, let go of notions of controlling others in order to maintain a semblance of Oh man, my brain. It's like going off too many tracks. I keep thinking too much about the abortion rights in the US, but I guess I just wanted to say that I think that interdisciplinary thinking is more important than ever now, because I think that so many of the things that surround us have so many dimensions to it.
Yeah. And I think respect really underlies a lot of that. Interdisciplinary collaboration is only ever founded on respect and not being territorial and not having a huge ego about what you're doing and trying to break down preconceptions. And that respect, I think, should know no bounds.
I love that. Thank you. That's a beautiful note to close on.
Yeah, it was a pleasure talking to you and being here.
Yes. And now we will turn it back to Veena.
Awesome. Thank you both so much. So many beautiful small golden nuggets of advice in there for the students as well. I appreciate you both for joining us.